Hello everyone, welcome back to A Lesson a Day. This is Nafis here, and today I'm yet again back in front of you with another interesting chapter of O-level biology, which is transport in plants. So, no more further delays, let's just begin with our lesson. Now, firstly, in this chapter, we will know about the structure of flowering plants. Now, highly evolved plants like flowering plants basically have a complex body, which may consist of fruits, leaves, stems, roots, flowers, etc. Now, these highly evolved plants can be classified as either herbaceous or woody. Now, herbaceous plants are those plants which have less amount of woody tissues in them, while woody plants are those plants which have higher amount of woody tissues or xylem tissues in them. Now, these plants can again further be classified into perennial, biennial, or annual. Now, you should know that when I say perennial, I mean they live for many years, and when I say biennial, I mean they just live for two growing seasons, and then when I say annual, I mean that they live or survive for a single year. Moving on ahead in this chapter, we are going to discuss how does transportation occur in flowering or highly evolved plants. Now, as you can see from the head topic, it occurs through vascular tissues. Now, what are vascular tissues? Vascular tissues are complex tissues responsible for transporting materials within a plant. Now, there are two types of vascular tissues, the xylem and phloem, and they have two different works. Now, basically, when we think about transportation, the two things that comes to our mind is that the manufactured food and water and minerals, which are basically getting transported around within the plant. Now, the xylem transport waters and minerals, and the phloem transports the manufactured food products. Now, let's get further ahead. Now, functions of the xylem. Now, xylem transport waters and minerals from the roots to the stems and the leaves, and also provides mechanical support to the plant. Now, talking about the structure of the xylem. Xylem is mainly made up of dead vessels. A vessel is a long hollow tube which is a continuous lumen stretching from the roots to the leaves. Now, as I mentioned before, it's a dead structure or dead vessel. I meant that it's made up of dead cells. Now, the walls of the xylem vessels are strengthened by a chemical substance called lignin. Now, lignin may get deposited in the form of spirals or rings, or in some cases, the whole vessel may get lignified, except the regions called pits. Now, in the picture down below, you can clearly see that how xylem vessels might get lignified. Now, moving on to do or perform the jobs that they are provided with, xylems might require some special features or adaptations, which they grow from the process of differentiation. Now, these adaptations mainly include um, absence of cross walls, which leads to a continuous path or lumen to carry waters and minerals. And another adaptation is that they have lignified walls, so that the vessels are strengthened to withstand the pressure of the water, and these lignified vessels further combinedly provide mechanical support. Now, many of you who are wondering what is a mechanical support is the support provided to withstand the harsh and extreme conditions by giving rigidity. Now, we are done with xylems. Now we are going to talk about phloems. Now, in one sentence, the function of the phloem is that it carries out the process of translocation. Now, but what is translocation? Now, translocation is the movement of manufactured food materials from the green parts of the plant, especially manufactured food from the green parts of the plant, especially the leaves, to the other parts of the body. Now, the manufactured food material includes sucrose and amino acids, etc. Now, the structure of the phloem will consist of the three main things: the sieve tubes, which will be made out of the sieve tube cells; the cross walls, which are perforated by minute pores and are like plates, so they are called sieve plates; and the companion cells, which are found beside every sieve tube cell. Now, moving on in our lesson, we will talk about some common misunderstandings and questions. Now, some people may ask, are the tubes of phloem are dead structures too, like xylem? No, actually they're not. The tubes are made up of living cells with degenerate protoplasm, though, so that the translocation can occur through the cytoplasm through diffusion. Now, why do they have companion cells? Now, as the sieve tube cells are degenerated, or they have degenerate protoplasm, the companion cell helps them to keep them alive. and also assist them in the process of translocation. Now, think it like this: they have many things missing. Like for example, they also have a missing nucleus. I mean, the sieve tube cells. So the companion cell has a large nucleus, so scientists sometimes think that companion cell sometimes also controls some activities of the sieve tube cell, and thus helping to keep them alive. Now, how can we prove that translocation takes place through phloem? We can prove it using AbFids or using carbon-14 isotope. Now, using AbFid, now the thing is, like, AbFid usually feed on plant juices. They use their proboscis to penetrate the leaf and the stem, but as the aphids leave, its proboscis gets cut off and stays inserted in the stem. A liquid may exude from the cut off part of the proboscis, which has been tested, and we have found out that it's been sucrose and amino acid. Now, after sectioning the stem and examining it, we have seen that the proboscis was inserted in the sieve tube of the phloem, thus making it as clear as possible that translocation occurs through the process of phloem, through the phloem tubes. Now, using carbon-14 isotope, this can also be proved. Like, for example, if you feed a plant with carbon-14 isotope, which is actually a radioactive isotope which can be detected on the X-ray film, the sugar that the plant will make will also contain the carbon-14 isotope. So, if we cut off a section of the stem and put it on the X-ray film, we will see that the radioactive isotope is being detected in the phloem, proving to us that phloem transports sugars and other food materials, thus it carries out the process of translocation. Now, moving on in our lesson, we are going to talk about internal structure of dicotyledonous root. Now, in a dicotyledonous root, the xylem and the phloem are not bundled together to form the vascular bundle. They lie on totally different radii, and the roots also have the cortex layer and the epidermis layer. And in the roots, the epidermis layer is basically bearing the root hair cells, and thus it's called the pelliferous layer. Now, talking about the internal structure of a dicotyledonous stem. Now, in a dicotyledonous stem, the xylem and the phloem tissues are grouped together to form the vascular bundle. The vascular bundle are arranged in a ring around the central region called the pith, and in the vascular bundle, the xylem and the phloem lie along the same radius, with the cambium tissue between them, which will further divide and form more new xylem and phloem tissues, increasing the thickness to the stem. The stem is covered with the epidermis layer, and the region between the epidermis and the vascular bundle is called the cortex. In the picture given below, you can clearly see each and every part labeled quite nicely. And one additional information I would like to add is that the cortex and the pith both serve as the storage materials or storage layer. Starch may be stored there. So that was it for today. Thanks for hearing me out, guys. I'll be back soon in front of you guys with another new video on this same lesson, or that will be the lesson 2 on this chapter. So until then, keep watching our videos on Lesson a Day.